Welcome, ladies and gentlemen. I was asked to talk about progesterone and its role and impact during pregnancy. And of course, I do that um, very often. For instance, also for a couple of pharmaceutical firms. That is the reason why I'd like to have this financial disclosure. Progesterone actually is a steroid hormones. And we have a couple of steroid hormones, and the parent compound is cholesterol. Cholesterol is quite an ancient substance. It could only be built, this typical ring structure, when oxygen tension on Earth was around 2 to 3%. And that is the reason why cholesterol, but also the steroid hormones, are practically present in every animal species. So steroid hormones are definitely quite important hormones. And today we are talking about progesterone. As you all know, progesterone needs a receptor to work inside a cell. And we have three different subtypes of receptors. Especially subtype A and B, both display a highly conserved DNA binding domain and a slightly less conserved ligand binding domain. The only difference between these two subtypes is an additional activation function 3 in progesterone receptor B. Progesterone receptor C is less well described. Interestingly, it doesn't have a DNA binding domain, and it's mainly present during pregnancy. The effect of this subtype is not yet clear. So when progesterone comes into the cell, it binds to its receptor, and then during the so-called classical genomic pathway is transferred directly to the DNA, to the so-called progesterone response element, and then activation of different cofactors is finally activating transcription of different genes. But this complex, the progesterone receptor complex, may also interact in a so-called non-genomic way to other structures, such as, for instance, tyrosine kinase receptors, and that initiates a MAP kinase pathway that finally leads also to transcriptional activities. However, this is a more rapid pathway. And finally, we also have a membrane-bound progesterone receptor, which also activates usually quite rapid intracellular metabolisms. In recent years, it became clear that steroids, especially progesterone, do not act via this classical pathway, binding to a hormone receptor and then binding to the response element and then leading to transcriptional activity. We know that different steroids, especially progesterone, may also exert epigenetic modifications. For instance, DNA methylation. There are effects that have been shown concerning histone acetylation, and even more recently, a post-transcriptional effect of progesterone and other steroid hormones is evident. The induction of so-called microRNAs. MicroRNAs are, as the word says, small pieces of RNA that may interfere with normal messenger RNA within the so-called RISC complex, the RNA-induced silencing complex. And when those kind of induced microRNAs are bound to normal messenger RNA, there might be a translational repression or even complete degradation of this messenger RNA, and that leads to an inhibition of mRNA to specific proteins. So obviously also post-transcriptionally, there are modalities concerning epigenetic modifications of effects. And these microRNAs are especially relevant also during a normal cycle within the endometrium, because RNAs are influencing different metabolisms such as inflammatory activities, apoptosis, angiogenesis, but mainly also cell proliferation and differentiation. And microRNAs may interfere with all these aspects. So actually, as you know, we have two different phases during a menstrual cycle. We have the estrogen-dominated proliferative phase, where a lot of cellular proliferation and growth and angiogenesis happens. And then after the LH surge, there is an increasing concentration of progesterone by the corpus luteum, and a specific ratio between progesterone and estrogen dominates the secretory phase, and especially around 6 to 10 days after the LH surge, there is an important window, the window of implantation, where the situation for an upcoming embryo is optimal to implant. So the main structures within the endometrium, the superficial layer, the epithelial cells, and the underlying stromal cells are influenced by steroid hormones in the first phase by estrogens, creating a specific estrogenic cytokine pattern with estrogen-darted genes, such as, for instance, the epidermal growth factor like growth factor. Once progesterone is increasing in concentration, there are inductions of specific signaling molecules, such as, for instance, the Indian hedgehog or HAND2, hard and neurograss derivatives, and specific target genes of progesterone, such as homeobox A10. And that all leads to specific modification of cytokines, not only within the epithelial cells, but also leading to stromal cell desidualization. And that is important for a receptive endometrium to lead to successful implantation. And as we also know, progesterone has a huge impact on the immune system, which I come back to that topic later in a couple of minutes. So the most obvious visible difference between the proliferative phase and the secretory phase is the immediate surface, the epithelial surface, which usually during the proliferative phase has a lot of so-called mucin. Mucin 1, these are small structures that keep microorganisms, for instance, but also the approaching embryo at a certain distance, whereas during the secretory phase, under the progestogenic effect, there is a complete reversal of this structure. So-called pinopods are erasing, and these are the interaction sites with the approaching embryo. So the increasing progesterone level, and to a certain extent also the cycle AMP level, leads to desidualization of the endometrial stromal cells via induction of different regulatory factors, HAND2, forkhead box 1, homeobox A10, or also signal transducent activator of transcription are upregulated by progesterone, and that leads to desidualization. And these cells then produce deciduous-specific factors, such as, for instance, prolactin or insulin-like growth factor binding proteins. And these decidualized cells do play important effects concerning the embryo. For instance, there is a loosening of the extracellular matrix by downregulating especially collagen type 4, and that helps the invading embryo to get access. There is a downregulation of radical oxygen species. There is also a change in angiogenesis, and there is an effect on the local so-called uterine natural killer cells, which is also important. Progesterone modulates the immune system of the mother because for successful pregnancy, it's important to do so because we actually have a semi-allogenic um, material. The fetus is partly maternal, partly paternal, and that and protective immune modulation is important. For instance, progesterone induces a lot more asymmetric antibodies that are non-cytotoxic and do not play any phagocytosis. Also, blocking antibodies are increased that mask fetal antigens. There is a switch from a more cellular immunity with a Th1 pattern to Th2 pattern, which is more a humoral immunity, and progesterone induces this uterine decidual natural killer cell activity, and that again helps this semi-allogenic fetus to implant. And there is one substance, the so-called progesterone-induced blocking factor, that is important in that respect because it induces all these immunologically relevant activities. So as you cannot investigate this situation in a human, usually in a mouse model is used because it resembles very much the human situation. Progesterone leads to decidualization, and that helps trophoblast invasion. Progesterone leads to epigenetic silencing of specific cytokine and chemokine genes, and that leads to a switch in the Th1 to Th2 pattern. Progesterone induces the progesterone-induced blocking factor, and that, for instance, has an immunosuppressive effect. Galectin, for instance, is upregulated, and this has been shown to arrest local dendritic cells. And progesterone also influences maternal immune tolerance, for instance, by upregulating the T regulatory cells within the endometrium, and there is also an enhanced recruitment of these cells, and altogether this leads to a specific immune modulation that is necessary for successful implantation. Dydrogesterone is a very interesting progesterone stereoisomer, a so-called retroprogesterone. It is actually the same molecule, but only one additional double bond between C6 and C7 leads to a certain bending of the plane of this molecule. And that in turn helps to be bound more specific to the progesterone receptor at the ligand binding domain, and that enhances dramatically the progestogenic effect of dydrogesterone. And in contrast to other synthetic progestogens, there is a very similar activity of dydrogesterone in comparison to progesterone. There is also an interesting effect. This bent molecule does not bind to the glucocorticoid steroid hormone receptor. So therefore, there is no glucocorticoid activity, which is, in my opinion, also a favorable effect. And this dydrogesterone can massively induce, for instance, progesterone-induced blocking factor during the early phases of pregnancy, and that seems to be quite an interesting effect. So clinically, where are our therapeutic indications of progestogens? We have three main areas. First of all, repeated abortion, recurrent pregnancy loss. It could be demonstrated in numerous studies that there is definitely a beneficial effect, as shown in this meta-analysis in 2017. The overall effect is significantly beneficial with progestogens. However, if you look into more details, it could be shown that these two large studies using dydrogesterone are actually the two studies that triggers this overall effect concerning beneficial in preventing recurrent pregnancy loss to a significant extent. There is also agreement that a support of the luteal phase by progestogens should be used in ART cycles. We know that with stimulation, usually estrogen concentrations are much increased in ART cycles, and therefore we need a counterpart, and that is the addition of progestogen. Again, meta-analysis have shown that this is of a significant advantage to add progesterone in contrast to placebo. You may also add beta-HCG. However, with that, you can reach better pregnancy rates, but there is also an increase in side effects such as, for instance, ovarian hyperstimulation syndrome. There is a very large prospective randomized trial concerning dydrogesterone taken orally versus 600 milligrams myconized vaginal progesterone. And actually, initially, this LOTUS trial was done as a non-inferiority trial, but then it turned out that there is at least a trend to a beneficial effect comparing myconized progesterone, not only with ongoing pregnancy rates, but also with live birth rates with dydrogesterone. It's not statistically significant, but there is a trend. Obviously, this pronounced progestogenic efficacy of dydrogesterone is beneficial. And finally, the prevention of preterm delivery by progestogens. We know that during a normal pregnancy, progesterone concentrations are rising sharply during the 40 weeks of gestation, and this high increase is actually produced by the placenta, by cholesterol, and we know that progesterone is absolutely necessary for uterine quiescence, for downregulating contractile proteins, not only the parent compound progesterone, but also metabolites such as 5-beta dehydroprogesterone. There are also other metabolites that are important as they react with the fetal GABA receptor in the brain. We know that may also have beneficial effects, but the main effect is downregulation of contractile proteins. And that again is obviously, as studies in recent years demonstrated, by the induction of microRNAs. Progesterone receptor complex induces SEP1 and 2. This is the zinc finger E-box binding homeobox, and that in turn leads to a sharp increase in the production of microRNAs, the family 200. And of these microRNAs, we know that they suppress contractile gene transcription, such as, for instance, oxytocin receptor, gap junction proteins, but also there is a decrease in the production of cyclooxygenase and therefore prostaglandins. So this is obviously the mechanism whereby progesterone leads to uterine quiescence. And we have 3 specific risk groups where clinically the therapeutic application of progestogens had proven effectively. First of all, women with a history of preterm delivery. The meta-analysis in 2016 clearly demonstrated that there is a favorable effect of progesterone, and more so in women with a short cervix. We know that a cervix cervical length below 2.5 centimeters at 24 weeks gestation is the most important relative risk factors, even more important than history of preterm birth or other factors. And in this collective, again, it could be proven that progestogen prevention is obvious in women with a short cervix until the 24th weeks of gestation. And that reduces preterm birth substantially from 22% down as low as 14%, with a very low number needed to treat. And this reduction in preterm birth is associated with a lot of reduction in morbidity and mortality of the neonates. There is a decrease in neonatal intensive care unit admission, a sharp decrease in respiratory distress syndrome, therefore a decrease in mechanical ventilation, and overall a decrease not only in morbidity but also in mortality using progestogens during pregnancies in these specific risk groups. And finally, also twin pregnancies with a short cervix below 2.5 centimeters have been demonstrated to have favorable results when supplied with progestogen during pregnancy. So in conclusion, we have three therapeutic indications where progestogens in the past have demonstrated beneficial effects. That is, in cases of repeated abortion, definitely as a support of the luteal phase in art cycles, and in specific risk groups concerning preterm delivery. Thank you very much for your attention.